Hi everyone, my name is Saad. The research article that I would like to present today is called Electrochemically Assisted Microbial Production of Hydrogen from Acetate. This paper was published in the year 2005 in the Environmental Science and Technology Journal. This paper explores a novel and innovative method for producing hydrogen gas using microbial fuel cells, MFCs, enhanced with an external voltage. This paper is divided into 5 different sections. First is the introduction, second is methods, and third is results, which is followed by discussion and conclusion. In the next slide, before starting to introduce the main paper, first I would like to highlight the important details, main contributions, and key points observed in this paper. As for important details, this paper shows the new method and usage of bio-electrochemical to produce hydrogen directly from organic matter like acetate. This process involves microbial fuel cells, MFCs, augmented by an external voltage. It uses acetate as an important byproduct of glucose fermentation and serves as a critical substrate in this study. It offers a direct route for microbial oxidation, releasing a high yield of electrons and protons, and also address the limitation of bacterial fermentation. For the key points sections, this paper significantly contributes to the reduction of the energy required to produce hydrogen compared to traditional methods such as water electrolysis. If we talk about water electrolysis, a theoretical minimum voltage of 1210 millivolt is required, and in practice, it often exceeds 1800-2000 millivolts due to overpotential. In contrast, the electrochemically assisted microbial fuel cell, MFC, system operates at voltages as low as 250 to 850 millivolts. This reduced voltage requirement is achieved by leveraging microbe metabolism to oxidize organic matter, and also it manages to utilize 90% of protons and electrons. Another key point is that this system is not restricted to glucose or carbohydrates like traditional fermentation processes. Microbial fuel cells can oxidize a wide variety of biodegradable organic materials, including acetate, as demonstrated in the study, other organic acids like butyrate, waste biomass such as proteins, amino acids, and wastewaters. Lastly, for the contributions part, this paper shows promising results because it demonstrates for the first time the direct production of hydrogen gas in microbial fuel cells, MFCs, by augmenting bacterial electrochemical activity with an external voltage. Traditional MFCs focus on generating electricity from organic matter, but this study shifts the paradigm by utilizing MFCs to produce hydrogen instead. Introduces 2 new reactor designs for hydrogen production, using MFCs, which holds different specifications and will be discussed in detail in the coming slides. Also, this broad substrate compatibility highlights the system's potential for treating various types of organic waste while producing renewable hydrogen. It significantly expands the applicability of MFC technology for sustainable energy production. Now moving to the next slide, which explains the introduction of this paper. In today's world, the need for a hydrogen-based economy is driven by the demand for cleaner and more sustainable energy sources to combat rising greenhouse gas emissions. Hydrogen is considered a promising alternative due to its potential for zero carbon emissions when used in fuel cells, but current hydrogen production heavily relies on fossil fuels, with natural gas and coal accounting for the majority of global output. Only 4% of hydrogen is produced through renewable methods, such as water electrolysis powered by solar or wind energy. However, to truly reduce carbon emissions, hydrogen must be produced from renewable and sustainable sources, such as biomass and organic waste, rather than relying on fossil fuels. Conventional chemical fermentation can generate hydrogen from biomass, but its yields are low, typically limited to 2 to 3 moles of hydrogen per mole of glucose. Even under ideal conditions, fermentation can only produce a maximum of 4 moles of hydrogen per mole of glucose when acetate is the primary byproduct. This inefficiency arises because acetate, a common fermentation byproduct, cannot be further converted into hydrogen without additional energy input. Consequently, much of the energy potential in organic matter remains untapped. The paper introduces the concept of electrochemically assisted microbial fuel cells, MFCs, as an innovative solution. MFCs utilize bacteria to oxidize organic matter, such as acetate, producing electrons, protons, and carbon dioxide. By applying a small external voltage, the electrons and protons generated by microbial metabolism can recombine at the cathode to produce hydrogen gas instead of water. This approach significantly reduces the energy requirement for hydrogen production compared to traditional water electrolysis, which typically requires 1800 to 2000 millivolts. In contrast, electrochemically assisted MFCs operate at much lower voltages, 250 to 850 millivolts, making the process more energy efficient. This whole process involves two key reactions, the anode and cathode reactions. The anode reaction involves microbial oxidation of acetate, generating protons and electrons from organic matter, while the cathode reaction combines these protons and electrons to produce hydrogen gas. Next is the methods section of this research article. The methods section of the paper details the experimental setup and procedures used to evaluate hydrogen production through electrochemically assisted microbial fuel cells, MFCs. Two different reactor designs were constructed, a two-bottle system and a compact cylindrical reactor, both featuring an anode and cathode chamber separated by a proton exchange membrane. The reactors used plain carbon cloth as the anode and carbon paper coated with platinum, 0.5 mg as the cathode. The anode chamber was inoculated with bacteria enriched from domestic wastewater, ensuring a microbial population capable of acetate oxidation. The anode chamber contained a phosphate buffer, 50 ml pH 7.0, and a nutrient medium, while the cathode chamber contained only the buffer. Both chambers were purged with nitrogen gas to maintain anaerobic conditions. In the two-bottle system, the chambers were connected through a clamped beam and filled with 200 milliliters of solution. Electrode spacing was 15 centimeters. The cathode chamber was sealed to prevent gas escape and enable periodic hydrogen sampling. In the cylindrical reactor, the electrode spacing was reduced to 0.25 centimeters from the PM to minimize internal resistance and enhance electron flow. Gas produced in the cathode was collected in a sealed 120 milliliter bottle for analysis. The applied voltage ranged from 250 millivolts to 850 millivolts using a programmable power supply, ensuring a steady flow of electrons. Acetate concentration was measured at the end of each experimental run using gas chromatography, while hydrogen gas production was quantified using a thermal conductivity detector. Coulombic efficiency was calculated by comparing the total measured coulombs to the theoretical maximum possible from acetate oxidation. This experimental setup allowed for the efficient testing of hydrogen production, enabling the evaluation of Coulombic efficiency, hydrogen recovery, and energy consumption in both reactor configurations. The methods ensured long-term stability and reproducibility of results, validating the feasibility of electrochemically assisted MFCs for renewable hydrogen production. Next is the results and discussion section of this paper. The results section of the paper presents findings from the experimental evaluation of hydrogen production using electrochemically assisted microbial fuel cells, MFCs. The study demonstrated that hydrogen production began when the applied voltage exceeded 250 millivolts, confirming the need for external voltage to drive the cathode reaction. As the applied voltage increased from 250 millivolts to 850 millivolts, both current density and hydrogen production rose significantly, indicating enhanced electron transfer efficiency at higher voltages. The hydrogen recovery from acetate oxidation exceeded 90%, while the Coulombic efficiency, the percentage of electrons recovered as hydrogen gas, ranged from 60% to 78%, depending on operating conditions. Acetate degradation was highly efficient, with approximately 95% of acetate consumed during each production cycle, suggesting stable microbial activity. This was followed by the discussion of the paper, which interprets the experimental findings and evaluates the feasibility of electrochemically assisted microbial fuel cells, MFCs, for hydrogen production. The researchers highlighted that hydrogen production began at an applied voltage of 250 millivolts, despite the theoretical minimum being 110 millivolts. This discrepancy is attributed to overpotentials caused by resistive losses, electrode surface limitations, and proton transfer inefficiencies, underscoring the importance of optimizing electrode materials and reducing internal resistance. The comparison of the two reactor designs revealed key trade-offs. The two-bottle reactor, with its larger electrode spacing, 15 centimeters, achieved higher hydrogen recovery rates, up to 92%, due to better hydrogen collection efficiency, but suffered from lower current densities due to increased resistance. In contrast, the cylindrical reactor, with 0.25 centimeters electrode spacing, achieved higher current densities, up to 7.1 amps per meter squared, but experienced lower hydrogen recovery, 60 to 73%, likely due to hydrogen gas diffusion into the anode chamber. The final part of this paper is based on the following factors. The conclusion of the paper emphasizes that microbial fuel cells, MFCs, can effectively produce hydrogen from acetate through electrochemically assisted processes, achieving high hydrogen recovery, 90%, and Coulombic efficiencies ranging from 60% to 78%. This demonstrates the system's ability to efficiently convert electrons generated by microbial metabolism into hydrogen gas. The study highlights that the energy input required for hydrogen production in this bio-electrochemical system is significantly lower than that required for conventional water electrolysis, where voltages typically exceed 1800 millivolts. In contrast, the MFC-based system operated with an applied voltage of only 250 to 850 millivolts, making it a far more energy-efficient and sustainable alternative. Another important conclusion is the system's substrate flexibility. Unlike traditional biological fermentation processes limited to glucose or carbohydrate-rich materials, this MFC system can utilize a wide range of organic substrates, including acetate, butyrate, and even complex waste streams, such as wastewater. This versatility positions the technology as a promising approach for renewable hydrogen production integrated with waste management and wastewater treatment. Lastly, the reactors demonstrated long-term stability during multi-month experiments, confirming the system's potential for continuous hydrogen production. These findings suggest that electrochemically assisted MFCs could become a practical, scalable technology for renewable energy generation, contributing to both clean hydrogen production and environmental sustainability. I hope that you enjoyed the informative and knowledgeable review about this research article. If you have any comments or questions, you can leave them below. Thank you for listening.